In this video, I'd like to talk about the process of lipolysis, which is essentially the breaking down of triglycerides into free fatty acids and glycerol. So let's write down this process. We have lipolysis, and essentially what we're doing here is we're taking our triacylglyceride, which is essentially the storage form of free fatty acids, and we're going to break it down into free fatty acids plus glycerol. Essentially, a triacylglyceride has these ester bonds, and we're going to break it down into 3 free fatty acids and a glycerol molecule, which forms the backbone of triacylglycerides. Now let's set the stage for this process. This process takes place when blood glucose levels are low. So low blood glucose is going to trigger this process of lipolysis. And this is going to lead to the release of a hormone, which is really important, and this hormone is known as glucagon. Specifically, your pancreas, your alpha cells of the pancreas, of the islets, can release glucagon when blood glucose is low. So when glucagon is released due to low blood glucose, it will stimulate this process of lipolysis. Other stimulators of this process include epinephrine and norepinephrine. So during a stress response, right, when you're in this fight or flight mode, what's going to happen is your adrenal medulla is going to release adrenaline or epinephrine. And this epinephrine can also stimulate this process, as well as norepinephrine. And this is due to acute stress from the sympathetic nervous system. In fact, most of what adrenal medulla releases is epinephrine, but also a little bit of norepinephrine. It's like an 80-20 split. So epinephrine and norepinephrine, due to acute stress, sympathetic nervous system, can also stimulate the lipolysis response. Other things that can stimulate it are cortisol, which are released from the zona fasciculata of the adrenal cortex, as well as the thyroid gland, which releases thyroid hormone. And also growth hormone from the anterior pituitary can also stimulate the breakdown of triacylglycerides into free fatty acids and glycerol lipolysis. So these are the stimulators, but the main ones that we will talk about, that we will use as examples in this video, include glucagon and epinephrine. So now let's take a look at how this process actually takes place. So here is an adipocyte, and this is essentially the adipose cell. And this adipose cell is going to have these receptors on it. And these receptors are GS, or G-protein coupled receptors. And these G-protein coupled receptors have 7 transmembrane domains, and they're called such because they're coupled to a G-stimulatory protein, to a GS protein. And in this process, you're going to form a second messenger. So let's actually draw that right here. So let's actually draw this G-protein coupled receptor right here. So we have this receptor, which has 7 transmembrane domains, 1, 2, 3, 4, 5, 6, and 7. And this receptor is going to have a site where the hormone can bind to. And in our case, we're going to look particularly at glucagon, but this can also be epinephrine or others, for example. So here's glucagon binding to the receptor of this transmembrane domain, and then, as you know, you're also going to have this GS right over here. So this is the G stimulatory portion of this receptor, and this is a G protein. And so what's going to happen when the glucagon binds, it activates this receptor. There's going to be a conformational change, which is going to cause the G stimulatory protein to bind GTP. So it's going to exchange GDP for GTP, and that's essentially going to make it active when it's GTP bound. And what's going to happen then, you're going to have the release of the alpha subunit of this G stimulatory protein, and that alpha subunit is going to activate a key enzyme on the membrane, which is known as, let's actually put this in a different color, let's make this yellow, adenylyl cyclase. I'm going to put a box for it right here, adenylyl cyclase. And this adenylyl cyclase is then going to convert, it's going to convert your ATP into cyclic AMP. So it's going to convert ATP into cyclic AMP. And cyclic AMP is your second messenger. And what's going to happen next is that this cyclic AMP is going to activate a very, very important protein. And this protein that it activates is known as protein kinase A. And this protein kinase A is going to put a bunch of phosphates on a variety of molecules. So it is essentially an effector. So again, when blood glucose levels are low, we have glucagon that's released, as well as epinephrine and norepinephrine, but we're looking at glucagon, which stimulates lipolysis. How does it do that? It binds to this receptor on the adipose tissue, which has 7 transmembrane domains right over here. And then this will lead to the activation of this G stimulatory protein, which causes the alpha subunit to dissociate and causes the formation of a second messenger, cyclic AMP. So the adenylyl cyclase will be activated and lead to the conversion of ATP to cyclic AMP. And the cyclic AMP will then cause the activation of protein kinase A. And what protein kinase A, what it will do is really, really interesting. So protein kinase A will actually add phosphates onto another molecule, also on the membrane, known as perilipin A. So let's draw that right over here. We have our perilipin A. So this perilipin is actually bound to another protein. And this perilipin that gets phosphorylated by protein kinase A is actually found on the triacylglyceride droplet. So when pKa phosphorylates perilipin A, this other protein that bound to it will come off. So let me show you a picture of what I mean. Here in the cell, right, we have our triglyceride droplet. So let's draw that right over here. So this is our triacylglyceride droplet. And on the membrane, like on the outer part of the lipid droplet, we have this perilipin A. And this perilipin A, let me write it down, perilipin A, is actually bound to another protein known as, this is a long one, CGI58. And so what protein kinase is going to do, protein kinase A will actually put phosphates onto this perilipin A molecule. So it's going to give phosphates to it right over here. And when it gives phosphates to it, what's going to happen next is the CGI58 will actually disassociate, let's do it in this color, will disassociate from the perilipin molecule. So now it's on it, so now it's by itself. And this CGI58, as well as the phosphorylated active, right? When it's phosphorylated, it's active. Perilipin A both play very, very important roles, which we will look at in a moment. Notice how on the membrane, or on the lipid droplet, we have these three scissors. 1, 2, and let's put one more right over here. 3. And as you guys know, inside you're going to have a triacylglyceride. This is a triacylglyceride droplet. There's going to be a bunch of them right inside, right over here. And so in order to liberate each of these fatty acids, we need to make 3 cuts. So let's look at exactly what's going to happen. So perilipin A is bound to CGI58 around the triacylglyceride droplet, but when protein kinase A phosphorylates perilipin A, the CGI58 comes off and stimulates the enzyme that makes the first cut of the ester bond to release the first free fatty acid. So we had this triacylglyceride with 3 fatty acid tails, and we're going to make that first cut in order to liberate a diacylglyceride, because we're going to cut right here this first ester bond. And the enzyme that's responsible for doing that is known as ATGL, or adipose triacylglyceride lipase. This is a lipase that's found on the adipose tissue, on the surface of the lipid droplet. This is the first scissor that I drew right here. And this is stimulated by that CGI58 that has been released. Also, the perilipin A that's been phosphorylated by the protein kinase A, the perilipin A also plays a crucial role. It actually rearranges the triacylglycerides in this lipid droplet to make the ester bond more accessible to the lipase enzymes. For example, the ATGL that we just saw, and also the other two. So it makes the triacylglycerides, it makes the ester bond where the cleavage occurs more accessible to the enzymes that cleave it, to the lipase enzymes. So that is really, really crucial. So the perilipin A also plays a very, very important role in this process. So let's also write it down. Right? It doesn't phosphorylate ATGL directly, but it allows it to access the ester bonds so that it can cleave it. And now you have the diacylglyceride. So remember, we had a tag, a triacylglyceride, and now we have a diacylglyceride. And then what's going to happen is it's going to get further cleaved. The next enzyme cuts the second ester bond. And now, again, we have this structure right here, but now it just has one fatty acid chain, and this is known as a monoacyl glyceride. I know I drew the glycerol as a huge one right here, but essentially we're left off with a monoacyl glyceride. And the enzyme that's responsible for converting this DAG to this MAG is known as, it's a very, very important enzyme, and it's known as hormone sensitive lipase. I'll abbreviate it as HSLD. a very, very important enzyme. And again, the perilipin A rearranges the triacylglyceride structure, so these enzymes can better access the ester bonds and cause the cleavage. But what's really, really interesting here is that pKa can actually directly phosphorylate this enzyme. So if you have this enzyme, the pKa directly phosphorylates and activates this hormone-sensitive lipase A. So this enzyme, the second one, actually gets directly phosphorylated by protein kinase A. Just like protein kinase A phosphorylated our perilipin, which causes this rearrangement, it also phosphorylates hormone-sensitive lipase. So now we have one more step. So this was our second scissors. Now our last one. This MAG is going to be converted to glycerol, and that last free fatty acid has been released, the third one, right? And this enzyme that's responsible for this conversion right over here is known as monoacylglyceride lipase, MAG lipase. This is the last enzyme. And again, the perilipin A phosphorylated active form helps rearrange the triacylglyceride, so this last scissor can access this last ester bond and liberate the final free fatty acid. Now, once we have the free fatty acid and glycerol, these can actually go to the blood. And the free fatty acid is not very water soluble. So let's have a look at it right here. The free fatty acid that I liberated, right? 1, 2, 3, in addition to the glycerol, are all going to travel to the bloodstream. Now the glycerol is water soluble, so you can easily get there. But the free fatty acids are not very water soluble. So what's going to happen? They're going to be bound to a protein known as albumin. So the free fatty acids are going to be bound to a protein known as albumin. And eventually, they're going to get to the target tissue, like the muscle, the heart, and the liver. And that's where the fatty acid is going to be oxidized to make ATP via a process known as beta oxidation, which we looked at previously. Now, I'd like to look at what happens to this glycerol backbone. So the glycerol backbone is going to actually go to the liver, and in the liver it can be taken up to make glucose via gluconeogenesis. Specifically, the glycerol will be converted to glycerol 3-phosphate. A phosphate group will be added to it, and the enzyme that will do this is known as glycerol kinase, which is present in the liver, in the hepatocytes. And this glycerol 3-phosphate can then be turned into another structure known as DHAP. And the DHAP can then be converted into another molecule, which is known as glyceraldehyde 3-phosphate. And this glyceraldehyde 3-phosphate can then go into two pathways. It can go into glycolysis to make ATP, or into gluconeogenesis to make glucose from a non-carbohydrate source, glycerol. So the free fatty acids that we actually burned at times of low glucose can make the free fatty acids which can be burned to make ATP in target tissues like the heart and the skeletal muscles and the liver, but the glycerol can also go to the liver to actually make these precursors to make glucose via gluconeogenesis. In fact, the fate of this molecule is determined by what the cell needs. So in this situation, when glucose levels are low, the gluconeogenesis will be favored and make glucose to raise the blood glucose levels, right? So if blood glucose levels are low, then we're gonna actually make more glucose from non-carbohydrate sources. And let me blow up a adipose tissue, and let's draw here a picture of the liver. So what's going to happen is in the adipose tissue, the fatty acids are actually going to be packaged into triacylglyceride, but as we mentioned, the triacylglyceride can also break them back into free fatty acid. So the free fatty acids can make triacylglyceride. It can go this way, but it can also go this way. And then the free fatty acids which are made in the adipocyte can then be carried by albumin and go into the liver, for example, and not to other vital tissues. And what's going to happen in the liver when blood glucose levels are low is the glucose will be taken up, undergo glycolysis, and eventually produce acetyl-CoA. And if you recall, that acetyl-CoA is a substrate to make free fatty acids. These free fatty acids can be packaged into triacylglycerides, right? So that's really, really important. And then these triacylglycerides can then be packaged along with cholesterol into very low density lipoprotein. This very low density lipoprotein is how we carry triacylglyceride. We can also carry it via chylomicrons, which are made in the small intestine, right, when we eat the fat. In this case, the liver makes these lipoproteins, VLDL, which can then go into various tissues, and via lipoprotein lipase, right, what's going to happen is that the triacylglycerides are going to be broken down into free fatty acids in adipocytes, which can then be repackaged into triacylglyceride. So the VLDLs contain the triacylglycerides, and they're like dogs that are going to bind to the surface of the adipocyte cell, and then there's going to be a lipoprotein lipase there, which is going to break these triacylglycerides in the VLDL into free fatty acids. And that's how we get these free fatty acids after we have eaten a lot of food, such as when blood glucose levels are high, so that they can be stored into triacylglyceride. But then when we want to mobilize them, we go the other way. So they're broken back to free fatty acids, as we saw in this video, and bound to albumin, because the fatty acid chains are not very water soluble, and they're then going to be used for energy. But also remember, this process also makes glycerol, and this glycerol can go to the liver for gluconeogenesis to bring the blood glucose levels up. So it can go both ways. When blood glucose levels are high, the free fatty acids that are made, packaged into VLDL and stored as triacylglycerides. And when blood glucose levels are low, what's going to happen is that triacylglyceride is going to be broken down to free fatty acids in the adipose tissue, and this will be mobilized to be used for energy in the liver, but also they're going to be sent to other tissues of the body, like the muscle and the heart. But also in this process, we make glycerol, which can also go into the liver for gluconeogenesis. So it can go both ways. Anyways, I hope you found this video helpful.